Hi, my name is Alexis Ellis. I go to American University in Washington, D.C. And this summer I was at Cornell University in Margaret Frank and Abraham Stokes Lab.
Would you tell us maybe what was the most important part of your project, some of the things that you did during your project and the major findings?
Yes. So my project was focused around a propydium iodine or PI staining. So typically in a PI stain, it involves taking a cell and moving it through different reagents. Each step takes about 20 minutes. So moving one solution, 20 minutes later, moving it again, 20 minutes later, moving it again. As you can imagine, this is very time consuming. This takes a lot of time out of people's day. And my project takes this process and it makes it automated. There are automated tissue processors out there. However, they cost anywhere from $10,000 to $15,000, which is very expensive for a lab to invest in. So a lot of plant scientists just end up doing this manually. The whole process takes about 2 to 3 days. My mentors made me go through it a lot of times, so I kind of get the struggle. So this takes that process and it makes it automated. Here I have my prototype, the first of many, hopefully. But we have some hardware components and we have some software components. So I can kind of go a little bit into depth about each one. So hardware wise, we have 3 peristaltic pumps. One tube is in our container here, and then our other tubes are brought into our premix container here. Demo wise, I just filled these up with color, but typically in a lab, this will be filled with ethanol and water. You would use 2 bottles to mix whatever percentage of ethanol and water you want. So let's say you wanted 15%, it would take some of one solution, take some of another solution, get you to that 15%. Our liquid would flow into here. This is our premix container. This container just makes sure that both the ethanol and the water are at the concentration that we need it to be. After, it'll flow into here. This is our tissue container where our actual tissue cells will be held. And then after, our ball valves will open up, and then it'll bring everything down to our big giant waste container down here. And that'll be disregarded after. These solenoid ball valves just make sure that everything can go from one container to another without leaks or anything. And then in the back, we have our Arduino and our relay. Our Arduino is actually where the magic happens. It controls all the pumps, all the valves, make sure everything goes when we need it to go. And then the relay keeps everything from running at the same time. So that's amazing. Software wise, oh my gosh. Okay, software wise, we have something called a graphical user interface. My graphical user interface utilizes 2 types of code. It utilizes Arduino code, and it utilizes Arduino code is in C++, and it utilizes Python coding, which is in VS Code. So sorry, it took a while, but here we have our graphical user interface. So this is what actually brings it up and makes it user friendly. This is what a plant scientist will use whenever they want to use this machine, because if you tell a plant scientist that whenever they have to use this machine, they have to go into the code, pick out the line, change that exact number variable, it's a little intimidating. So this just makes sure that it's user friendly towards them. And then the actual magic and the codes that the commands that the Arduino is receiving is all in here. This is where all the math comes in about the flow rates. All of that is done in here. So we go here and we press play. It'll bring up our graphical user interface, and it makes it user friendly. So it'll ask the number of steps. We'll just put in 2 and then continue. And then in my original code, it'll ask for the percentage of ethanol, and it'll bring up anything from 15% to 100%. But for the purposes of this demo, I've changed all of the percentages to colors. And then you go here and you can pick whatever color you want. So we can do orange and we can do purple. And then once you press initialize, that's when the plant scientist job is done, and you don't have to worry about anything else until the program is done.
That's very reassuring.
Yes. So then it takes a lot of the weight off of a plant scientist. And then it goes, and then this program will fill up to the 100 line in about a minute, and then it'll flow into our tissue container here, and then it'll wait 20 minutes there. Well, no, this code will immediately go from here down to our waste container, whereas in the original code for the PI staining, it'll fill up to this 200 line mark, which takes about 2 minutes, 2 to 3 minutes. And then it'll flow into here, and then it'll wait 20 minutes here. So that way the cell can be hydrated or dehydrated. And then after 20 minutes, it'll bring everything from here to our waste container.
Oh my gosh.
Yes.
So fun.
I think so too. I had such an amazing time. My mentors were absolutely amazing. My PIs were absolutely amazing. So social.
Yeah.
And I feel like I've had overall a really great summer. I was able to bring in like my engineering background as well as fulfill my love for that.
Yeah,
So
I love that. I think it's so important, the overlap, the interdisciplinary part
I
of
agree.
science is so crucial to making great advancements like this, which is a great way to save money in the lab and
yes.
time and energy.
And then overall, this only cost about $250. We did have some money go into these, which is machine shop by our machine shop engineer in the lab.
Yeah.
But $250 compared to $10,000 is still pretty affordable.
That's fantastic. Well, what was your favorite part of all of this, and what was new to you? Because it seems like you might have already had a hand in the coding and everything like that. What was maybe the most challenging new aspect of this project?
Honestly, the code, because I've worked with code before, but not to this caliber. I've done Arduino stuff, but just like little stuff like flashing lights, like, oh, like that's cool.
Yeah.
But definitely nothing to this extent. I would say the most rewarding part is also the code too, because there's something so fulfilling when you like see everything finally work for the first time in the way and in the manner that you intended it to. Yes.
Cool. My goodness. So do you think that this is applicable to other, protocols in labs?
Yes. So even though this is made for a typical PI stain, I've had other people come up to me and they're like, yeah, I could totally use this in my lab just for even like watering plants. Or someone came up and said, oh, this would be great for mixing drinks. And I was like, I guess.
Crescent party trick.
Yeah. This will soon be an open source for other labs to replicate. So I think that is also going to be very helpful.
That is, congratulations.
Thank you.
Really fun project. Thank you so much for sharing.
Thank you.
Last details you want to share? Anything else you want to share?
This will be implemented in the Frank lab in the fall for future testing.
Perfect.
So
Do you have a name for it?
I do. Okay, so this is a coffee machine that we stole that we borrowed from the third lounge of our hall. So I was like, yeah, this would be perfect for our bottles. So once we got it, its name, like I guess the trademark on this was Mind Reader. So I decided to name it Mind Reader. I think that was really cute.
That is fantastic. That is excellent. Well, thank you again so much. And I hope you had fun.
Thank you so much for coming to speak with me. I really appreciate it.